Bismillahirrahmanirrahim, and welcome back to this next video. And this is yet another video in the series of videos on the bioinformatics. In my previous video, I've told you how you can turn a DNA sequence into a protein, and I've told you that if you know the sequence of a DNA, you can translate it into the corresponding protein sequence by using the genetic code. And the genetic code is the very same thing that the cell itself used to generate a protein sequence from a given DNA sequence. And this genetic code is universal, and it's nature's solution to the problem of how one uniquely relates a 4 nucleotide sequence to a suite of the 20 amino acids. So what is this particular genetic code? So in this particular video, I want to focus on the genetic code table. If you look at this particular table, there are 64 codons if you count them. So the whole of the codon, they are 64, and they code for 20 amino acids. What I mean by a codon is the combination of 3 nucleotides. So if you look at this one, this is one codon, this is one nucleotide, this is another one, this is another one, and when these 3 nucleotides combine with each other, they are going to make a codon, and this codon is going to code for an amino acid. For example, this UUU is coding for the phenylalanine, like this CCU is coding for the proline, this CAU is coding for the histidine, and so on. So how this genetic table works, how you are going to find out the sequence of a particular codon. If you can see over here, this particular row or this particular column is showing you the first letter of the codon. This is going to show you the second letter of the codon, and this particular area is going to show you the third letter of the codon. So you have got the U, C, A, and G as the first letter. The U, C, A, and G then is the second letter of a codon, and then this particular area is going to show you the third letter of the codon. So how are you going to make this? If you look at this one, this is U, so this would be the first letter for this particular codon, so it is U over here. The second letter again is coming from this particular area over here, again this is U, and if you look at the third letter, again this is U. So this is U, U, and U, as you can see over here, and this UUU is actually coding for the phenylalanine. If you talk about the second codon, this UUC, it's also code for phenylalanine. So how this sequence of the codon comes? As you can see over here, the first letter again is U, the second letter is U, but if you look at the third area or the third letter, this is C over here. So this is U, U, and C, and again this U, U, and C is coding for the phenylalanine. If I give you another example, for example, from this second area, so the first letter is C over here, so this is C. The second letter again is coming is U over here, and if you look at the first letter over here, this is U. So this CUU is coding for the leucine. So I hope you are getting the idea how this particular genetic table work. Let me make it a little bit complex from the second or from the third letter. For example, if you look at this particular area, so the first letter is the C, and if you look at the second letter, that again is this would be A, and as you are talking about the second area, this second one, so again this is going to be the C, because the C is corresponding over here, and if you look at the first one, this would be U, because this is U in the third letter area. So the CAU or the CAC, this is actually coding for the histidine. So this is how the genetic table code works, and in this particular table you can see you have got 20 amino acids over here, and you have got 64 codons. Now 61 of these codons, they code for the amino acids, and 3 of the codons, they are stop codon. And as you can see over here, this UAA, this is the stop codon, this UAG, this is the stop codon, and this UGA, that is the stop codon. What I mean by the stop codon is that whenever the translation machinery it reads these particular stop area or this particular stop codon, they are going to stop the synthesis of the protein, or that would be a signal that no more protein should be synthesized, because this is coding for a stop codon and not for the amino acid. So 61 of the codons, that will be coding for different amino acids, and 3 of the codons, they are going to code for the stop, and that means stop the protein synthesis. So if you like the video, please subscribe to my channel, hit the like button and share it with your friends.